Hello, this is the Unit 2, Module 6, Chapter 13D presentation. The big picture coverage for this last section of Chapter 13 describes the cultivation of viruses and concludes with the presentation of the non-viral infectious agents, the viroids and the prions. Viruses can be cultivated within suitable hosts, such as a living cell. They are placed with a cell type that the virus, under cultivation, is able to infect. Before cell culture techniques were developed, fertile hens' eggs were used to cultivate viruses in the laboratory. If the cells showed changes, known as cytopathic effects, then the culture is positive and the virus is growing. Cytopathic effects refer to damage to host cells during virus invasion. Degenerative changes in cells can also be studied in tissue culture. This damage is measurable by obtaining so-called viral titers. Viral titers can be assayed, for example, by looking for plaques using a monolayer of tissue culture cells. Direct counts can be ascertained using observation and the electron microscope. A quantal assay involves a series of dilutions that will yield the infectious dose, which means the virus is able to infect 50% of the cells to which it is applied, as well as being able to yield the lethal dose, the amount of virus that when added to a quantifiable number of cells will kill 50% of the cells upon infection. We've talked about bacterial-specific viruses and animal cell viruses. Plant cell viruses are also extremely common, and although they are common, no plant viruses are known to infect human beings. Viroids are plant pathogens. They consist of a short stretch, usually a few hundred nucleobases, of highly complementary, circular, single-stranded RNA. They have no capsid associated with them. The human pathogen hepatitis D virus is similar in structure to a viroid. Finally, there are particles that are even smaller than viruses or viroids, and they are infectious and cause disease. These are the prions. Prions are misfolded proteins containing no nucleic acid. They cause transmissible spongiform encephalopathies. These are fatal neurodegenerative diseases and are unfortunately common in animals. The disease scrapie in sheep and goats is a wasting disease that is caused by prions. Bovine spongiform encephalopathies, also known as mad cow disease, are caused by prions. And in humans, Creutzfeldt-Jakob syndrome, or CJS, is a prion disease. Scrapie and normal uninfected cow brains are in this slide. The scrapie-affected brain, infected with prions, is on the left. The normal cow brain is on the right, and the differences in pathology are obvious. Transmissible spongiform encephalopathies can occur in humans and animals. TSEs are neurologic degenerative diseases, and they're easily transmissible by the consumption of tissue that has been infected with a prion. Originally, scientists believed that TSEs were caused by viruses, but Stanley Prusiner discovered the prion, which is short for a proteinaceous infectious product. The death of a host that has been infected with a prion occurs from nerve cell death, which leads to sponge-like holes in brain tissues. The symptoms of a prion encephalopathy include dementia, weakened muscles, and a loss of balance. This results from the formation of insoluble aggregates of abnormal prions in the brain. Brain shrinkage and deterioration occurs very rapidly. The disease itself, in humans, as shown on this timeline, can take up to 30 years to manifest itself after the initial infection. Prions themselves are resistant to a wide range of chemical and physical treatments, such as formaldehyde, ultraviolet radiation, and heat up to 80 degrees centigrade. This makes them extremely resistant to the usual sterilization techniques. Prions have a normal cognate or form in cells. Proteases readily destroy overproduction of this normal cellular cognate. But the infectious prion form comes into a cell and takes the normal cognate and as a template acts to refold the normal cellular protein, making it become insoluble and able to form aggregates. The replication of prions is complex, but the summary is that it involves these mutant prion proteins converting normal cellular proteins to mutant prion form proteins. The modes of transmission for prions include injection, which can occur during a blood transfusion, ingestion of infected tissue, which can occur, for example, if humans consume meat that is already tainted and infected with prion proteins, the transplantation of contaminated tissue, for example, kidneys or corneas, from a donor that has been infected with a prion encephalopathy. And finally, contact with contaminated medical devices, brain electrodes, for example, that have been inserted into a person who has been infected with CJD, and now that CJD prion can be transferred to another person via the contaminated electrode. This slide contains questions to reflect upon and use the presented information. Use the learning objectives for Chapter 13 to guide your study of the information from both the presentation and the textbook. Be sure to define all the vocabulary terms. Take the Module 6 vocabulary test. When you are ready, take the Chapter 13 test. This concludes the Unit 2, Module 6, Chapter 13 presentation.